Welcome. This is Dr. Kirk Hollander once again, and we'll be talking about genetics for the third week in a row. This week we'll be talking about genetics and its relationship with inheritance. This first lecture, or part one, we'll be talking about some general background, some terms as key phrases and terms and concepts that we'll be using to then expand upon in part two and part three. In those lectures, we'll be focusing on the genetic basis of inheritance, how genes and traits are passed down, and then we'll also be talking about some associated diseases. So with that, I hope you enjoy the lecture, and here we go. So as always, we have objectives. Our first objective will be to understand how locus and allele relate to homozygous and heterozygous. These are really four basic concepts that really set the foundation for everything we're going to talk about. The second objective will be to explain the relationship between genotype and phenotype, another very concrete and necessary terms that we need to expand upon for our future discussions. Then we'll talk about how to use a Punnett square and understand the use of a pedigree chart. And then finally, we'll describe penetrance and variable expression and how they relate to our observation of traits and potential diseases. So I wanted to begin by trying to tie in what we talked about previously with this slide in our discussion of mutations, and then try to link it into what we're going to talk about in genetics and eventually leading to disease. So here is our outline of the different potential mutations that we observed previously. So we had our unedited or unchanged original sequence of template DNA, original mRNA, which then translated into our polypeptides that make up proteins. And then we talked about how we have these four different types of mutations and how they lead to changes in the polypeptide sequence, which eventually lead to changes in the function of the protein, often causing large defects in the protein, allowing it to be non-functional. We also talked about gain of function, as well as the loss of function, like we mentioned, with the loss of protein folding correctly. And so what we're going to talk about today is how these changes and mutations are described based off where they're located and what alleles, and how these are then passed on to subsequent generations and the pattern of which that proceeds. So with that, we're going to try to move on to our next slide, talking about specific terms associated with the DNA sequence and how we describe that. So to genetically discuss these potential mutations we talked about, we need to lay the groundwork of some specific terms. So in this slide, I want to talk about locus and alleles. So a locus is a specific location on a chromosome. And so what's interesting about this is that there's conservation of these genes on the chromosome. And what I mean by that is that specific genes map to specific chromosomes, and they map to very specific spots on chromosomes. So if we have homologous chromosomes here, like one from in the paternal homologue pairing with the maternal homologue, they will have paired genes at that specific spot on those chromosomes, right? So they're like copies of each other, right? Those homologs, which would be copies, they're at this locus or this location, there is a gene encoding for P. And in the maternal one, there is at that same location another gene encoding for P, all right? Now you may have also noticed that there are different, there's a capitalized P on the paternal homologue and a lowercase p on the maternal homologue. And so what this is it's indicating different versions of this gene, all right? So the different versions of the gene could be examples of slight alterations in that gene causing specific changes to the protein or causing the different changes to the expression of that protein. There are different forms of genes, and these alternative forms can have great effects that we'll talk about in the next few slides, but it's really just specific alteration or different versions. So we're talking about allele, we're talking about a version of that gene, and we're talking about one allele, we're talking about allele on one homolog. If we're talking about both alleles, we're talking on both homologs and how it works together. Now, the other basic concept we should discuss here is homozygous or heterozygous. So, homozygous would be if the two copies of the gene are very much the same. So, a genotype in which two copies of a gene determine a particular trait are the same. So, our example here would be Y. If you look at the locus Y, right, that location, we have an uppercase Y here and a lowercase y here. That is how in genetics we would indicate that they are the same version. If they are both uppercase Y, those would be the exact same copies of the gene. And so when we have the exact same copies, we're homozygous, where they're the same copies on both homologous chromosomes. You see, we have that paired uppercase Y, uppercase Y. Conversely, heterozygous means you have two different alleles for that. You have two different alleles for a gene that determines a trait are different. So you'd have different outcomes from that locus. And so this would be indicated here, but you can see you have the uppercase P and the lowercase p, right? This would be a heterozygous genotype, and you have two different versions of the alleles at the P locus, all right? So homozygous would be the same allele at the same locus. Heterozygous would be different alleles at the same locus, all right? And that's kind of how they all work together. Locus, allele, homozygous, heterozygous. So when we're talking about locuses and on chromosomes and the different mutations and the DNA code that we observed, what we're really talking about is the genotype. And the genotype is the actual DNA sequence. When I was doing research on mice, I was looking for mice that were fitting a specific genotype that I was trying to drive and create in them. I would physically read the DNA code based off of some advanced techniques, and then that would be the genotype of the mice. So that would be the actual DNA sequence of the mice leading to their genotype. Now, conversely, we have phenotypes. So phenotypes are observable characteristics. So for example, here we have eye color. You can see blue, green, brown, other shades of brown. These are observable characteristics. These are phenotypes. These are things that you can physically see with the eye, or sometimes things that you can measure. We often call these things traits. So a phenotype is an observable trait. And so we have this correlation and relationship between genotype, the DNA sequence, which eventually leads to a phenotype. So in research, we're often looking to see if we can correlate a genotype or a sequence that we find and then see if it matches up to a phenotype that we can observe. So there's this balance and relationship between the two. But it really just wants you to know that genotype is the actual DNA sequence and the phenotype is that observable characteristic or trait. Now, an example of some observable traits that you may be familiar with would be like a cleft chin or no cleft, or people either have dimples or not. And so we have these different observation or phenotypes we can observe. But what's interesting is that there's a relationship between how often we can observe different phenotypes, all right? So people could either have a cleft chin or no cleft. And what's been found or observed is that more people often have a cleft chin. And we call this the dominant trait, in that there are more people in the population having it, and so then we associate that with dominance. Recessive would be that they're less observed, and so no cleft in this case, and so those are the least or lesser observed, or the least, there's less likelihood of that being observed in an individual. Now, when we link these traits, the dominant or recessive traits, we can link them to the alleles, all right? So the version of the gene. So the dominant allele will express its phenotype regardless of the instructions of another allele, which means that if you have the allele for cleft chin, even if you had the other allele for the no cleft, you would always see the cleft chin, all right? that cleft chin allele is dominant over the recessive in that it masks this recessive allele. Another example would be dimples, right? Dimples, people with dimples, it's a dominant trait. More people observe with dimples. If you have one allele for dimples, it will mask the other allele that does not have dimples. So you will always see people with, you don't always see people with dimples, but people carrying the dimples gene would observe dimples even if they only had one allele. Now you will only see these recessive alleles if they're expressed in, if they are having the recessive allele in 2 copies, right? So both alleles would have to be recessive, and then you would observe these recessive alleles. So it's only the chance that we would observe recessive allele if you don't have a dominant allele at all. So to rehash, the dominant allele will express its phenotype regardless of the instructions of the other alleles. So regardless of if you only have one allele that are dominant or 2, you will see the dominant allele. If you don't have any dominant alleles and you only have recessive alleles, then you would observe the recessive phenotype. All right, and we'll talk about how we kind of map this out a little bit more in the next slide. And so we map this out with Punnett squares. So some of you may remember this genetic tool from your past, but it is a kind of a mapping or diagramming tool that we use to predict and also then kind of follow what's going on within our dominant or recessive alleles and how they interact. So what a Punnett square can do is you can show all the potential offspring from a specific cross. And so what we have here is we have the dominant alleles being expressed by the uppercase T. Lowercase t is representing the recessive allele. And so what you can have is you have the parental alleles either here across the top or across the bottom. And so in this case, this is a monohybrid cross of these 2 parents. We have one parent with a recessive, or excuse me, a dominant and then a recessive allele, and then this parent has 2 recessive alleles. So we do this cross. What I do is I would start with the uppercase T and I'd put it in this square and then this square. And then I would start with the recessive allele from the parent up here and put it in this square and this square. And then you fill out the other way. So you take this lowercase t, you put lowercase t here, recessive allele here, recessive allele here as well. So when you map this out, you have 2 homozygous recessive offspring right here, or a 50% chance of a homozygous recessive offspring. But you could also have a 50% chance of a dominant allele with a recessive allele. So you would have a heterozygous individual showing the dominant trait because the dominant trait masks the recessive trait as observed here. Now, when we have 2 kind of different types of crosses that we do in this we can use for testing, and it's been genetics. If you go back to the individual pea pod experiments, it was based off of these monohybrid or dihybrid crosses. And what you're really doing is you're looking at hybrids being crossed, and the parents are only differing in one trait in the monohybrid, similar to what's here, and you end up with this 3-to-1 ratio. You end up with a 3-to-1 ratio demonstrating the relationship of the 2 different hybrids crossing. In more complex, you can end up with a dihybrid cross where you're looking at 2 different traits and seeing how they crosses. If it's a standard heterozygous across the board, you could end up with a 9-to-3 to 3-to-1 ratio, indicating that the observed genotypes would be in a 9-to-3 to 3-to-1 relationship. But we're not going to go into a ton of detail about these crosses, specifically these test crosses. I just wanted to kind of tie it back to maybe some of the stuff you've had in the past. So don't really focus too much on these monohybrid or dihybrid crosses, just more kind of for demonstration purposes. But in the next slide, I do want to talk a little bit and kind of review how you do set up these Punnett squares to kind of make sense what we talk about later in the lectures. So we're going to go back to our Punnett square and kind of tie it into eye color, which will help maybe, hopefully helps explain this kind of Punnett squares for review, I'm sure, for many of you. So we have our monohybrid cross here, as alluded to in the previous slide. So we have two individuals with brown eyes, and they're crossing. We're going to look at their offspring, all right? Now, these individuals with brown eyes were heterozygous. So we have one large allele, or excuse me, one dominant allele, B for brown. So the dominant allele is brown. Blue is a recessive trait. So blue, lowercase b, is the recessive allele, all right? So when we do this cross, once again, I like to fill in from the top down. From this parent, I put a B in the upper left quadrant and the lower left quadrant, the uppercase B. And then the lowercase b, I would then fill into the upper right quadrant to the lower right quadrant, right? So you're just filling down, right? B, large B, uppercase B, uppercase B, lowercase b, lowercase b, lowercase b. All right, then across the, from left to right, you take that uppercase B, you'd fill it here, then you'd also fill in uppercase B here. You take the lowercase b here, and you'd fill it in there, and also in here. All right, so you're just moving left to right, filling in. Now, as a result, you get the resulting combination of offspring showing their different alleles. So 25% or 1/4 of the offspring would be dominant, have the dominant or brown eyes and be homozygous, right? Because they have both alleles are the dominant gene. 50% would be heterozygous, but expressing the dominant allele, right? Because the dominant allele is expressive over the heterozygous, so they'd be brown. So you would observe heterozygous showing the dominant allele, so brown there. And then the final quarter, or 25% of the population, you would observe the blue eyes, because you have both of the recessive alleles are in that individual. They have none of the dominant alleles. So in the absence of dominant alleles, you will see the recessive alleles expressed, and you'll see the recessive phenotype, which would be blue. So in this case, you're getting that 3:1 ratio, which I previously mentioned on the test or the monohybrid cross. Now, if you had that individual with blue eyes in the next generation was crossed then again with an individual who had brown eyes, but also carried the recessive allele, right? So they are heterozygous, and they're being crossed with the homozygous recessive. If you fill out this Punnett square as we did previously, so you're filling out down and filling in across, you will end up with the following genotypes. And as you can see, 50% of this population is showing the homozygous recessive genotype, and is also showing the homozygous recessive phenotype, or the blue eyes, right? So you can see they received both recessive alleles and ended up with the blue eyes. You also then, due to the dominant allele, you have dominant alleles, you're seeing the heterozygous individuals showing the brown eyes because they have the one dominant allele and the one recessive allele in both that. So you have a 50% chance in this cross of the homozygous recessive being crossed with a heterozygous, indicating you're going to get the 50% chance of individuals with blue eyes. So this is kind of just a walkthrough of how you set up Punnett squares and how you go through them. And really, I think should emphasize how the dominant allele does mask what you're observing in the recessive allele and leads to these representative populations you get where the recessive allele is much less represented in the overall population. So Punnett squares are a good way to label out and observe the chances of populations giving rise to different phenotypes. But there's another way that geneticists use, and actually it's widely used in clinician practice or genetic counseling, in that it's called pedigree analysis, and that's a way to kind of map family trees, genetic features, to then explore and see what's happening through multiple generations. What you're trying to do with these is tied into a specific pattern of inheritance, which we'll talk about in the next few lectures. But I just wanted to introduce pedigree analysis to kind of give you an overview so you kind of know what's going on before we get into the patterns of inheritance. So we have our key up here. So males are represented by squares, females are represented by circles. If an individual is affected, most often these pedigree analyses are used and associated with some sort of disease, or at least or chance of disease, and so we talk about in terms of affected individuals. And so affected individuals then are colored in or fully filled in shapes, either male or female. So you can see they're completely filled in. We describe mating relationships by horizontal lines connecting the males and females together, and then we talk about their offspring. As you can see, an offshoot or a product of this mating, you have a horizontal line showing these siblings. So you have the male here and its two sisters. And if we start tying it in, we can see that this one male is affected, and his sister is also affected. And then we could looking at multi-generational, right? So in this case, this male then mated with with a female, leading to the next generation of 3 boys and 2 girls. And in this generation, 1 female was affected and 1 male was affected. And then they also have their subsequent marriages or matings, as you can see here down the line. So you have this multiple generations. Males are always squares, females are always circles, and the affected individual is always filled in. There is instances where it is skipping a generation, and then sometimes they will put a dot in the middle of the either square or circle to show that that individual carries a trait but doesn't manifest or show the actual disease or the trait we're looking at. But we'll talk about that more in detail when we get to an actual example in the patterns of inheritance. So there's 2 other terms that I'd like to talk about, which is penetrance and then expressivity. So we'll start with penetrance. So penetrance is how often a trait or condition is observed within a present population. So just because an individual carries a trait, as I alluded to before, they don't always express the phenotype that we would expect or is not observed. And so you have within a population, let's say this is a population where they're all carrying our specific genotype. And so in complete penetrance, they would all express the phenotype of the affected individual. So it could be some disease, right? They'd all showing this disease. However, in reality, in genetics in real life, it doesn't always come out to be 100%. So an example is we get incomplete penetrance or reduced penetrance, which means that there are individuals within the population that carry the genotype that do not express or show the disease. They don't show the disease phenotype. And so this is the incomplete penetrance. The disease is not penetrating fully through the population, and so you have decreased amount of the population that is showing the disease. And so it's not in all, the disease is not being shown, or you're not getting symptoms within all those individuals. Now these individuals still carry the genotype, and so they can pass it on to subsequent generations. So they still carriers of the disease, but they're not showing the actual, they're not showing fully the phenotype as we'd expect. Now an example of this is retinoblastoma, which we'll talk about in the next slide. So retinoblastoma is a cancer of the eye. You can see this affected individual here showing issues in the retina and starting to have the full dilation of the pupil, eventually leading to loss of sight, unfortunately. This cancer begins in the retina and will spread. It follows an autosomal dominant pattern. We'll talk about that in a little bit and what that exactly means, but just to kind of give you context if you're coming back to reviewing this, just for completion's sake, it is an autosomal dominant pattern. Now on this pedigrees, you can see here, now we're getting to that dot which I just mentioned, all right? So we can see that we have affected males indicated here, showing the dark we have the dark squares. Now we also have squares with a dot or circles with a dot. That dot is showing individuals that are carriers of the genotype, so they have the genotype, but they are not expressing the disease state that we would expect. And so when you see that dot, that's what they mean. In retinoblastoma, it's a familial, it's carried on, it's passed from generation to generation. And so we kind of talk about it in terms of families. Less than 10% of families that have this retinoblastoma within their family line have this low penetrance phenotype, in that not all the individuals will show the disease or the affected state. This is a small population of the families that have, in most cases, people with retinoblastoma do have this higher penetrance where almost all the individuals are being affected by this specific disease. But this is a good example of reduced penetrance in general in these individuals, of 10% of families that have a low penetrance phenotype associated with it. Finally, we talked about, I want to tie in then variable expression. So penetrance was if the individual is expressing it or not. Within that range, we have what's called variable expression. So this means the severity of the disease can vary for individuals carrying the same disease, all right? So if the individual is carrier of that genotype, in their expression of that disease or that affected state, there is variability in how severe it is, all right? And so this can be affected by a few different things. So the environment, right? What that individual is taking in or what it is being exposed to, right, can lead to more or less severity of the disease that they're being associated with. So let's say, for example, if somebody was had a better chance of being type 2 diabetic, for example, right? If they were on a poor diet, you would get much more severity of their diabetes, and so would lead to a much more problem. It's not the best example, but just kind of, I wanted to say that the environment does play a level into the severity of diseases, and that's the same thing that's happening with these individuals carrying these genetic diseases. There's also modifier genes. We talk about genes as these simple inheritance patterns, but everything's much more complex than that. There are other genes that filter into this and cause changes to happen, leading to either suppressing the phenotype by some sort of checkpoint pathway, or either amplifying it due to you have two different defects within that along the same pathway, leading to different disease. So the genetic the genetic background or the other genes in play or the other alleles in play can affect the severity of the defect. And then there's also allogenic heterogeneity, which means that on a specific gene, you could have an issue where you'd have multiple mutations within the same gene. And my example, this would be like beta globin can lead to either sickle cell anemia or beta thalassemia. And so these sickle cell and beta thalassemia are all mutations on beta globin, but they are different mutations. And they lead to different severity of disease, even though you are being affected at a very similar locus on the same gene. So there's heterogeneity in being affected, that specific gene, just due to the type of mutation at either a different spot, which leads to different severity of a different type, slight variation of a different disease. Now, penetrance and expression work together, and they're kind of very commonly interwound. So we have on the left side, or on the top here, of our wild type phenotype as the circled non-filled in. The mutant phenotype, similar to our affected individuals, is a filled in oval. And we showed here before we had the complete penetrance, where it's completely penetrant. Everything, all these individuals are being affected. In incomplete penetrance, once again, you have these individuals where they are, they're not all being affected, so the penetrance of the disease isn't complete through the whole population. Now, if we tie in expressivity, we have narrow expressivity in both of these examples, in that it's either full on or full off. It's either there or not. And so there's a narrow expressivity. That expressivity is very tightly tied into the if you carry the or express disease. It's like an on-off switch, completely on or completely off if you're showing the affected disease state or trait. Now, conversely, down here We have complete penetrance, similar in this sense, where all these individuals are showing some level of disease state. So the disease is completely penetrating. And in this state, conversely, we have the incomplete penetrance, similar to above, where they are not all being affected. Some individuals are not showing the disease within them. Now, to contrast the top to the bottom, we have a broad expressivity. So what this means is we have different shades of how much the disease is affecting them. And so this is kind of like the dimmer switch, right? It's either slightly on or slightly off, or there's a range at which the disease state could be shown in that individual. So the top here was like on off, right? Switch on and off. The bottom here in the broad expressivity is you have a dimmer switch. We have that large range. And so this really affects how severe or not severe, and you can kind of go through this whole spectrum showing you have this very broad range of expressivity. And so these penetrance and expression tie in together and can really give you a much more complex and really truthful representing the actual population, actual disease states, showing how it plays in and how that affects the population as a whole. So I just want to quick rehash our objectives as I like to do. We talked about how locus and alleles, the different spots or different variations, relate to homozygous, being they have the same allele, or heterozygous where they have different alleles. We talked about genotype, the actual DNA sequence, and how that can then relate to a phenotype or an observable trait. We walked through Punnett squares and how they work, and kind of how you can fill them in. And then we went into talking a little about pedigree charts. We're looking at multi-generational families and how they're affected. And then finally, I talked about penetrance and variable expression, showing how penetrance shows how much a disease is within a population, and then you have variable expression with how different severity of the disease is within certain individuals. I hope you enjoyed the lecture, and I will see you in the next lecture.